Okay, I'm doing two things here. I'm demonstrating what we're about to write on the board. I'm taking a sip of this coffee that's full of sugar. That was a pretty big gulp. I hope that's enough to get me all the way through the lecture, but if not, I can come back here and take another gulp. Let's draw what's going on inside my body. And maybe to give us our first little visual, we've got an animal cell up here now. So unlike the plant, we don't see any chloroplast in here. And like the plant, we still see these mitochondria. There's a bunch of these mitochondria in animal cells, and let's draw one up on the board. As always, I don't really like this bar. I'm just going to ignore it, and we're going to put, kind of like we did with the chloroplast, let's put an organelle up here. This is our mitochondria. Okay, hopefully we can see that. Then this mitochondria, when we look closely, we're going to see it's a double membraned organelle. So I'm going to put another one of these membranes . Okay, let me label those. Now we've got some compartments up here. Like we can see in this cell, all of the fluid that the mitochondria is floating in, that's the fluid of the cell. We call that the cytoplasm. And then we're going to have these two membranes of the mitochondria. Let's label this one the outer membrane. And obviously this one will be the inner membrane. There's a lot of stuff embedded in this inner membrane, and I'm not going to draw it yet, but let's label these two compartments either outside of the inner membrane or inside the inner membrane. This space inside is known as the mitochondrial matrix. So the matrix is within the inner membrane, and then in between the inner membrane, this is what we call intermembrane space. Intermembrane space, like interstates, they run in between states, and so this is the space in between the two membranes. Okay, so cytoplasm outside the mitochondria, mitochondrial matrix deep within, and then you have this intermembrane space. We haven't put anything in the inner membrane yet, but we've labeled the compartments. And anytime we get into this mitochondria, let me just put another side note about the mitochondria. What this thing is really going to be doing is right off the bat, as soon as we get in the mitochondria, it's going to be using oxygen. So it's going to be using oxygen to separate carbons. We're going to see these chains of carbons are going to be broken apart in the mitochondria, and every time we break a chain of carbon into a smaller piece, we're going to see oxygen is used to separate those carbons. It makes carbon dioxide. I tried to put an overview of our formula that we're looking at today. This is the opposite of photosynthesis that we looked at. In photosynthesis, we started with sunlight, water, and carbon dioxide, and we ended up with oxygen and glucose. Here we're doing the opposite. We're going from glucose and a little bit of oxygen. The oxygen comes in when we start to involve the mitochondria. Maybe that's worth a review question real quick. If we did have a bunch of oxygen gas out here in high concentration, how is this oxygen gas going to get into this mitochondria? Simple diffusion. Right, it's tiny, it's nonpolar. So oxygen gas can just diffuse right in. It doesn't need any type of special channel. This is our simple diffusion. And we remember that carbon dioxide is going to behave that same way. It can just cross the membrane wherever it wants to. Okay, so we're going to be using oxygen here, and we're going to end up making carbon dioxide. And like we just pointed out, carbon dioxide, wherever it ends up being made, is going to be able to just cross through the membrane by simple diffusion, just like oxygen did. And then this is also something kind of weird. At the end of this, at some point, we're going to end up making some water molecules. And this is kind of the opposite of photosynthesis. Remember, we were using the sunlight to bust up water molecules last time, and now we're going to end up making a water molecule. We could do a bunch of review things. Remember when we busted up a water molecule, there were things like hydrogen ions, oxygens, and electrons that were coming off. We're gonna end up seeing that again, except we're gonna piece together a water molecule instead of breaking it apart. Are we ready for we doing okay so far with this? All right. I am going to put a glucose molecule up on the board. I'm going to take over this little middle space and let this be cytoplasm as well. That'll give me a little bit more space. And out here in the cytoplasm, we're going to see a glucose molecule. I'll write it as its chemical formula first. We've got 6 carbons, we've got 12 hydrogens, and we've got 6 oxygens. I sometimes draw this as a hexagon shape and label it our glucose. But maybe most helpful for this diagram, I'm just going to take these 6 carbons and focus on those. So let's take 6 of these carbons, this little 6-carbon chain, and let's see how it gets chopped up into individual glucose. Let's try this again. We're taking this one glucose molecule and we're going to see how it gets chopped up into individual carbon dioxides. So we're going from large to small here. And it's going to take place in a series of steps. The first break in this glucose molecule is going to happen out here in the cytoplasm before we ever get into the mitochondria. So let's see if I can have just enough space to draw this. This step right here, where the glucose molecule has just been broken into 2. I'm just going to use the space up here to write, this is glycolysis. Glycolysis, glyco, that's just our glucose, and then lysis is to split. All we're doing is splitting a glucose molecule, so this process of glycolysis happens outside of the mitochondria. And since we're not involving the mitochondria, it doesn't require any oxygen. So this is a single step, a single karate chop into the glucose molecule that yields 2 of these 3-carbon structures. We've got 1 6-carbon, we karate chop it into 2 of these 3-carbon structures. These 3-carbon structures get a different name. Instead of calling it glucose, we're going to call these pyruvate. And we've got 2 of those. So 1 glucose molecule gives us 2 pyruvates. Okay, and then there's something else that's come into mind when we were talking about going from large to small like this. There's things that are coming off. Energy is given off. We've unlocked a chemical bond. So some of that energy is going to be used in converting our dead battery here into a charged up battery, ATP. Same thing down here. We're going to see one of these ADP molecules is going to be converted into ATP. And the other thing, remember, I'll just put this in a darker green color. Since we're not doing any chloroplast, I'm going to change my green color to represent those things that were coming off. When we broke these chemical bonds, there were those electrons and those hydrogens that were coming off. We called those free radicals. So down here, we're also going to see those free radicals being released when we karate chop this glucose molecule. We saw those in photosynthesis, and also in photosynthesis, there was a molecule that was picking those free radicals up. We had talked about this NADP molecule before, so P was involved in photosynthesis. Now that we're not talking about photosynthesis, we're just talking about what happens in the mitochondria, we can leave off that P. But this NAD molecule is an electron carrier. It's picking up these electrons and hydrogens, and when it picks them up, we now write it as NADH. So it's loaded up those electrons and hydrogens. The same thing down here. We're going to see that this NAD plus molecule gets saturated with these electrons and hydrogens. So NADH. We've seen this before, but remember these were at the end of an electron transfer chain, and they were picking up those electrons at the end of the staircase. Here we can see, now we're doing a catabolic reaction. We're breaking chemical bonds. So every time we break one of these bonds, we're going to have free radicals released, and some of that energy is going to be used to quickly form an ATP molecule. So if we're keeping track so far, we have not involved the mitochondria, we have not involved any oxygen, and we've made, I'll just come down here to keep track, 2 of these ATP molecules. So no oxygen, small amounts of ATP. We've ended up with 2 of these pyruvate molecules. And it is actually these pyruvate molecules that the mitochondria is going to feed on. So any questions about the diagram so far? Before we jump into the mitochondria, we've got the 2 spaces labeled in there. Everybody good? Okay. I didn't leave myself a whole lot of room in this matrix, but let's see what happens when each of these pyruvates is taken into the mitochondria. I'll take this one first. Let's pull it over. When this makes its way into the mitochondria, we're going to see right off the bat, let me make that a little bit longer. We're going to see right off that one of these carbons is going to get ripped off. And then there's a 2-carbon chain that's left over. And I'm going to temporarily draw on one of those carbons. I'll just put a little asterisk next to it. We'll show this twice. So each pyruvate that's pulled into the matrix of this mitochondria, we can see one carbon is ripped off. And what happens to that carbon is an oxygen is going to get added to it. We've just made a carbon dioxide molecule. Like we said, the mitochondria is immediately going to start using oxygen to separate carbons. The same thing happens with the other pyruvate down here. When this pyruvate makes its way into the mitochondria, a carbon is going to get ripped off, paired with oxygen to make a carbon dioxide, and we're left with this 2-carbon chain. The reason I'm putting a little asterisk on here is that this 3-carbon chain of pyruvate, it gets modified into this 2-carbon structure, which let's just label, this is what we're going to call acetyl coenzyme A. The little asterisk is my way of abbreviating that there's a coenzyme attached here. So acetyl, I'll just put Co-A. There's 2 of these acetyl coenzyme A molecules made for each glucose. So the glucose is broken down to pyruvate. This pyruvate enters the mitochondrial matrix. We've already made 2 of these carbon dioxide molecules, and you know what's going to happen if carbon dioxide starts to build up too much in here. It's just going to be able to diffuse right out through the membranes. Just like oxygen came in, carbon dioxide can diffuse out. The thing that I wanted to add here, it is true that in this rearrangement from pyruvate to acetyl coenzyme A, there are some of these electrons and hydrogens that are released. So even down here, a couple of electrons and hydrogens that are released. Anytime we see that, the same, it's worth putting up on the board again, that same molecule is there to wrangle up those electrons and hydrogens. So NAD plus is going to get converted to this NADH. Put it up here too. Okay, we haven't talked about, we can see them picking up electrons, but we haven't talked about where they're taking them yet. So let's investigate a little bit further. There's no ATP made at this step because we've rearranged pyruvate to acetyl coenzyme A. It's still this 3-carbon structure. There's a carbon in that asterisk. So now that we're inside this mitochondrial matrix and we've got this acetyl coenzyme A, the process that happens in the middle here is what's called Krebs cycle. And like we were seeing in photosynthesis, Sometimes these processes have multiple names. Krebs cycle is also known as the citric acid cycle. You can use either name, Krebs cycle or citric acid cycle. What these cycles start with, they start with this 2-carbon structure, this acetyl coenzyme A. And as each turn of this citric acid cycle goes, this 2-carbon structure is going to be ripped apart into 2 carbon dioxides. So you can imagine if both of these acetyl coenzyme A's go through here, we're going to end up with 4 carbon dioxide. Now we've got 6 carbon dioxides in here. We know that these are not going to stay built up. Carbon dioxide can just diffuse out by simple diffusion. Thank God, now I have more room to write. Okay. So the Krebs cycle, by the end of the Krebs cycle, you can see that our entire chain of glucose has been broken down into individual carbon dioxides. So once again, every time we break one of these chemical bonds, here we've gone from a 2-carbon structure down to individual carbons. So what is also coming off of this, let me get my colors right, all along the Krebs cycle, we're going to see electrons and hydrogens being released. That means that the job of this NAD, it looks like a P, let me make that more like a D. NAD plus, like we said, is picking up those electrons, going to NADH. There's one other thing that I wanted to add since we've gotten so far without pulling down any of these slides. I'm going to remove citric acid cycle. Also, I'll leave Krebs cycle up. I just need more room to write. So this Krebs cycle, when we're breaking these bonds that hold carbons together, we know that these free radicals are going to come off. So in the other space, I'll write electrons and hydrogens. And I'm doing so just to compare the fact that there is another one of these electron carriers, just like NAD, except it gets a different name. We're going to call this FAD with a little plus sign. FAD acts just like NAD, and it's going to pick up these electrons and hydrogen. So when it does, we're going to draw it as FADH. We know what it's doing. It's picking up those electrons. It's about to carry them somewhere. So we're just about to the part of the drawing where we need to add where those electrons are going. And let me see if we're doing okay on each of the steps. We've made 2 ATP so far. We rearranged pyruvate to get to this acetyl coenzyme A, released some free radicals in the process. Then these 2 carbon acetyl coenzyme A molecules are fed into the Krebs cycle. There's one more thing I need to add there. Since we are going from a large to small molecule, that bond that we're breaking is not only releasing the free radicals, it's also going to be used to make an ATP molecule. So the dead battery of ADP, the energy from breaking this bond, is going to be used partly to stick a phosphate onto that ADP. And we have 2 of these, so even though I'm not going to draw this twice, we know 2 acetyl coenzyme A molecules are made per glucose molecule. So this happens twice. We would end up making 2 of these ATP molecules just by breaking the bonds in glucose. That's 4 ATP so far. That's not a lot of ATP molecules. There's something that I think is kind of magical that happens in between these membranes of a mitochondria that yields like 34 ATP molecules. And what's so genius about it is it's the same technology that we've used to get electricity to Vegas, right? These hydroelectric power plants. Instead of water flowing, spinning a turbine, we've got flowing ions across a membrane. Let's put those things now that ions were flowing across. I'll try and use, let's put this red color up, because we used this last time to represent our electron transport chains. I'll abbreviate electron transport chain. And there's tons of these electron transport chains. I'll get lazy and I'll just draw these 4. Right? But we're going to see these electron transport chains all along this inner membrane. Right, these were the staircases that we mentioned last time. We saw these electron transport chains in between photosystems. I know in my brain I've switched over to photosynthesis real quick. Remember what was hooked up on either side of these electron transport chains in photosynthesis was a photosystem that was catching photons of light and getting those electrons excited to the top of the staircase. We don't have that here. Right? So the question is, how do those electrons get to the top of the staircase? And here comes the answer. It's been sitting on the board all part of the time, waiting for us, these electron carriers. We've got two different electron carriers, NAD. It turns out NAD is going to be the high energy electron carrier. It's going to drop its electrons right up here at the top step. So these electrons, as they fall down the steps, are going to be used to do work. They're going to be doing the same work that they did in the chloroplast, so we'll review that in just a second. But NAD drops its electrons at the top of the staircase. Look at this thing. We've got FAD over here that is also carrying electrons to the staircase, but FAD is carrying lower energy electrons. So these are going to be dropped into the electron transport chain, but not all the way at the top step. So they're still going to fall down the electron transport chain. It's still going to be used to do the same work. But these are lower energy electrons, so they don't fall through as many steps. So they're not going to do as much work as these high energy electrons. I just point that out because later on we'll see that the mitochondria usually makes around 34 or 36 ATP. And I was always like, well, why is it 34 or 36? It depends on how many of those electrons are high energy electrons, or if you got some low energy electrons, you might not make as much ATP. Most of the ATP is going to be made the same way it was made in the chloroplast, which is the help of this ATP synthase. We were putting our ATP synthase up in a purple color. I'm going to fit one in right here. This we were putting as our ATP synthase. And these ATP synthases are going to be, they look like the tree structures. These are going to be open towards the matrix side. And if I had enough space, I would write, all along this inner membrane, we would see multiple ATP synthases all facing this inner matrix. That's a clue as to what direction these hydrogen ions are going to be flowing. We know how these ATP synthases work. Remember, they take the dead battery of ADP, and as long as these things are spinning, they're going to create ATP. But this wheel isn't going to spin on its own. There has to be something moving across it. So before we get to the things that are moving across it, let's pick up at the last of the story. We just talked about how these electrons get to the top of the staircase, or at least get halfway up the staircase in FAD's situation. The previous story that we talked about, electrons moving down the electron transport chain, in photosynthesis, it was this NADPH that was ready to catch them. And that's not the case in mitochondria. Remember, mitochondria are going to end up making water. When we busted up a water molecule, we needed electrons, we needed hydrogens, and we needed some oxygen. We yielded electrons, hydrogens, and oxygen when we busted up a water molecule. If we go down here, we're about to put together a water molecule. The last acceptor of this electron, as we get to the end of this electron transport chain, in mitochondria is going to be an oxygen. So electrons and oxygens are going to pair together with some hydrogens that are present. And at the end of these electron transport chains, we're going to see a water molecule is formed. So oxygen is the last electron acceptor. It's going to be paired together with hydrogens and make these water molecules at the end of each one. So we can see how we're making water. Those electrons at the end of the electron transport chains are pairing with oxygen and hydrogens, and we're getting water molecules off of that. So now we can see where electrons start and then where they end up at the end of that electron transport chain. And now let's mention what they're doing as they fall down the steps. It's the same thing they were doing in the chloroplast. Electrons, as they're moving down these electron transport chains, Because it's not really like a series of steps. We said in reality, it's a series of proteins that I've drawn here as a series of circles. And as electrons are moving down those series of proteins, we said they're grabbing hydrogen ions from an area of high concentration, and let me try that again. These electron transport chains, they grab hydrogens from a low concentration. I was about to draw this in the wrong direction. We know what's happening down electron transport chains. Hydrogens are being pulled across the membrane. And they're always going to be pulled from an area, they're going against their concentration gradient here. It turns out the intermembrane space is going to have a very high concentration of hydrogen ions. It might be helpful just to write this out. The matrix is going to have a very low concentration of hydrogen ions. Now why is there so few hydrogen ions in the matrix? And the reason is because each of these electron transport chains are like vacuums. They're throwing hydrogen ions to this intermembrane space. So the concentration of hydrogen ions inside the matrix is very low. They're constantly being thrown to the outside. So that's why we have such a high concentration in the intermembrane space and such a low concentration inside. You can imagine if the hydrogen ion concentration gets so high out here, then when those hydrogen ions get close to the ATP synthase, they're going to naturally want to flow inside, which is what they do. And as they move in, they spin that turbine, they generate ATP, and they don't stay in here for very long, because as long as those electrons are in these bouncing down those electron transport chains, the hydrogen is thrown right back out. And that's the trick, right? If we can keep a bunch of hydrogen out here, more so than in this matrix, then hydrogen is naturally going to want to flow in to this inner matrix and spin this turbine of the ATP synthase. I don't know where I have room to write this, but the movement of hydrogen through the ATP synthase, this magical step right here, is what is going to result in somewhere between 32 and 34 ATP molecules. And that's significant. I mean, this is not the only one. There's multiple ATP synthase proteins embedded in this inner matrix. I've gotten lazy and drawn just one of them, but they all work the same. And this is really the magical step where most of that ATP is produced. By far, way more than just the 2 ATP that are produced in glycolysis out here in the cytoplasm. There's even, like we said, 2 ATP produced by breaking down that glucose, but the majority of the ATP comes from setting up this imbalance of a bunch of hydrogen ions on one side of the membrane, very few on the other, and taking advantage of the flow of ions across the membrane. You can see anytime we involve the mitochondria, we're using oxygen at some point, and that carbon dioxide that builds up is able to diffuse right out. We know electrons have to be fed down those electron transport chains. They pull hydrogens to one side of the membrane, where those hydrogens want to flow back to the opposite side through those ATP synthases. Okay, you likely will see a review diagram of this in a week. It's not something that you're going to have to draw on the exam, but I can tell you if you do take the exam and use the blank side on the back of it and sketch something like this, it will help you navigate a bunch of those multiple choice questions.